and some leftover strawberry oatmeal bread that I made. And then I ate a yogurt. So no real vegetables or anything.
So you'll make a couple of
We had dinner from so I just put everything that was in the
Oh, I made it hot.
you have something? In fact, I have something that I want to talk to you about. How we're supposed to have your thing,
Oh no, I'm just like being
Just being a perfectionist.
totally.
Good.
I saw that. I think that would be a
But I wanted to give just a quick introduction to a couple of people. Thanks for coming out. Two orders of business first. If you have a lab and you've seen that there's a notice out that you have to do an electronic filing of your chemical inventory list, we have to keep our chemical inventory list up to date anyway. We've been doing it paper copies like on an annual basis. They're moving to an electronic system. And it's due actually on Friday, so if you can, try to do it. I know your lab has done it, thank you for that. The nutrient analysis lab is in the process. So if you have a lab, do the electronic filing, because we're just required to keep updated list for emergency purposes. Second to that, once those are in, we'll also have a series of criteria that EH&S will review for your labs. Once you pass that criteria, and I'll send it out to you, I'm still working on it, then you'll be allowed to order hazardous materials with your credit cards again. So right now we can't, we're kind of writing POs on that. And that's because we were out of compliance with the comptroller's rule that they need to be able to track hazardous materials. And they weren't able to track it with credit cards. So so everything has to be ordered through a PO, but if your lab is authorized after going through this process, then you can use credit cards to purchase hazardous materials. So I'll get that out to you probably today or tomorrow, and then if you comply, it lighten that burden up. Well, my other announcement is pretty soon it's going to be his problem to deal with all this. I'd like to introduce Dr. Todd Crowell. He's going to be the new director of CERC starting relatively soon, sometime March, April-ish, right? So, you want to say anything else? So, welcome to FIU and CERC. So, you want me to? Okay, well, we are honored to have the eminent scholar, Professor Evelyn Geiser, of our own CERC in the biology department. And she'll be talking on advancing limnological theory through global links in the global links in the ecological observatory network.
Thank you. Well, yeah, I'm going to be talking about some different kind of stuff today. I suppose you've heard me talk about the Global Lakes Ecological Observatory Network a few times in the past, but it's been a while since I've given you an update on what we're up to in this program and what we're doing here at FIU relevant to GLION. And I'm going to give you a bunch of little snippets of stories. I usually like to give talks that are like one long story or whatever, but we're doing a whole bunch of different things in the context of GLION, and I wanted to give you a feel for what a global network can be and some of the reasons why I've enjoyed so much working with GLION. And I realize that we don't necessarily have a bunch of limnologists in here, so I think one way to come at this material is to think about whether it would be worthwhile in whatever discipline you're in to be a part of something bigger nationally or internationally. I've found that this program has boosted my science and my career in a way that I hadn't suspected or anticipated, and that I've really, really enjoyed. And so that's the take home for this talk. That, you know, I think if we all had one of these kinds of networks for each of our disciplines, our science would be really, you know, rocking the world right now. So this is what I want to go through. What is GLEON? So a little overview of what this network's all about, a little bit about why I got involved in it, and some updates on the stories that you have heard about if you've come to my talks in the past about what we're doing up at Lake Annie at Archibald Biological Station. A lot of my work with GLEON is happening in the context of a working group called the Theory Group, where we're testing some general, really basic questions, primarily about phytoplankton assembly. And I'll talk a little bit about that and how Lake Annie fits in and some of the next steps in that work. And then end with, you know, again, why working within a larger network can be really rewarding and also really fun. So what is GLEON? We put together, the sort of organizers within the network, a little sort of horse and pony show about what GLEON is. So I've grabbed a few slides from that. These are just really general overview slides of what the program's about. And of course, we all know that our water resources are under pressures that we have not experienced to this degree in the past. So there's a big need to understand what's happening in our freshwaters in order to properly handle these kinds of problems going on. At the same time, the advancements that have been happening in cyber infrastructure and in sensing technology have been happening, you know, faster than ever before as well. And so here we have on the right, that's Verge and Jude up on Lake Mendota, you know, doing their early limnological sampling with the good old Schindler trap that weighs about 3 tons as you drag it through the water column. And, you know, that's what we think of in classical limnology is we go out and we drag a plankton net through the water and we take some water samples and measure the chemistry and whatnot. Well, now we have throughout our LPR sites, and this is definitely not just specific to lakes or oceans or terrestrial landscapes, but, you know, throughout our different kinds of habitats, we're able now to sense environmental change with different kinds of machinery that can measure phenomena at scales that we haven't dealt with before, so on the order of minutes, you know, to seconds. And so that gives us this new kind of window. It's as if everything in the past, we've been trying to understand ecosystems with our eyes closed most of the time and open once a month when we go out and sample, you know, and now we're, you know, at least seeing things in every, you know, few minute intervals with these kinds of sensors. So what GLEON is up to is really merging this idea of really having a bigger need than ever to understand what's going on in our freshwater resources with all these advancements that are happening with technology, and bringing them together to really trying to advance limnological science. And so here's our mission. It's kind of wordy, but we're conducting innovative science by sharing and interpreting high-resolution sensor data to understand, predict, and communicate the role and response of lakes in a really quickly changing environment. And we're going about this, we think about GLEON as a network of 3 things. And the first thing is that it's a network of people, and this is probably the most, well, it is the most important aspect of any network, I think, is the people who are in it. The people who are in it are self-selecting. They want to be collaborating with other people to learn more. And in GLEON, we've seen this really rapid growth. We started around 2006, and you can see there the trajectory and the number of members. And the distribution around around the world. And I just put there on the side that the leaders currently, or well, Tim Kratz from University of Wisconsin, GLEON was kind of his brainchild. He had been working with a bunch of folks at the I forget what it's called, the big computer center in California, and trying to integrate what they were doing in Wisconsin on lakes with the kinds of technology that have been coming out of the computer center in California. And so, you know, this was a bit of his brainchild, and now Kathy Weathers at Carey Institute and Paul Hansen at University of Wisconsin are the co-chairs, but we have distributed leadership from throughout the world, pretty much representatives on a steering committee from all the different countries. So that's first and foremost a network of people who like thinking about different aspects of this kind of science, from the technological all the way to the, you know, limnological theory. We're also a network of observatories, and this is constantly growing as well. Formally, we have about 60 lakes that are sending data to this GLIAN network. There are a lot more lakes. Pretty much everybody who's a part of GLIAN has some lake that they're working with that may not be a formal member in terms of making data broadly available to everyone. I'll talk a little bit more about that challenge a little bit later. And finally, we are a network of data. So as you can imagine, if you think of all of our classical data sets that are sometimes difficult to deal with, if we're collecting information over years or decades, that's a lot of information. But if you do the same thing and your information's coming in every 15 minutes, then it's even more. So we have a lot of data streaming in from a lot of, these are our monitoring buoys. Here's the one at Lake Annie, and here's a whole bunch of different ones around the world. And these are all streaming numbers on a huge variety of parameters. I think I have a list here. Yeah, some of the common things, most of these are associated with the weather station so that we can understand climatic variability. Most of them have temperature profilers to understand water column thermodynamics. Dissolved oxygen data are always being telemetered somehow to a central station. We're looking at things now with sensors that can pick up different kinds of pigments, so we can measure abundances of different groups of algae and things like the amount of color-dissolved organic material. These are all classic limnological variables that we now have sensors for. We can stick them out on buoys and understand their high-resolution dynamics. So that's GLIAN in a nutshell, a network of people, of observatories, and of lots and lots of data. This network, again, it started up in about 2005, 2006. How did I get involved in it? Well, you probably know I've been working up at Archbold Biological Station for a while on the weekends and holidays. I love going up there, and I hope you all have some experiences up there. It's a wonderful place to work. And Lake Annie is kind of a holy grail of for the paleohistory and as a sort of sentinel of climate variability for Florida, because it is on this protected property of Archbold Biological Station, so it doesn't have a whole lot of, you know, major land use change going on around it like all the other lakes in our state, and it has this really deep limnological record, paleolimnological record, like 9 meters of sediments representing 40,000 years. So it's a really cool lake to work on. Here's a little just images of where it's located. So we're up there northwest of Lake Okeechobee. It's about 2 hours and 15-minute drive from here. It's the lowermost lake on the Lake Wales Ridge, which is all full of wonderful sinkhole lakes. So these are all dolines, and they're really deep and really beautiful, and at the head of the Everglades watershed. This is the headwater lake of the Everglades watershed. It's at the highest point on the Lake Wales Ridge, and all the water from there is going through this chain of lakes, eventually into Okeechobee and down. So we can use Lake Annie as sort of a sentinel of what to expect in our watershed down here. It's at the head of it is a tiny little lake, only 37 hectares, but it's 21 meters deep, so this is classical sinkhole feature. It's in this kind of scrub vegetation with a big bay head in the corner. This is a beautiful LiDAR image of the sort of contours around the lake. And it is mostly groundwater fed, so it's in a big sand, you know, all the substrate up there is all, you know, it's really deep sand. It's like 200 feet of sand. And so the groundwater moves through that really freely. And it's a monomictic lake. Do you guys remember what monomictic means? So it's mixing once a year in the wintertime and stratifying because it's so deep throughout the rest of the summer, spring, summer, fall. And the cool thing about it is, as I said, it has this incredible paleolimnological history going back some 40,000 years. That's what it's most known for. When Bill Watts started working there in the 1980s and discovered the whole, that was the first reconstruction of climate variability for the state of, or for the southeastern United States, really. It's how we understand Holocene history in this region. And then we have manual observations that remarkably go back 30 years, and this is kind of a rare thing too. So there were some real visionaries. There's always been visionary directors at this place. Jim Lane said, hey, we've got this treasure right here that is, not messed up by orange or citrus groves and, all the other kinds of things. Lots of people living around it. And it could be a really good sentinel for change, and so we should start studying it. And there was a postdoc there at the time, Larry Bateau, who in 1983 started working on the lake and saying, okay, let's go out monthly. And this was all, it is all volunteer. There are volunteers that go out there. The education coordinator's been going out or she went out for 25 years until she retired taking these samples. And now there's another guy who's doing it once a month. So we have an incredible manual observational record. And that got kind of handed over to me because there aren't any limnologists working up there, and I started going up there and found out about it, and Hillary Swain, the current director, said, why don't you take this over, and that happened in about 2001. And this is what we see. This is a really, really cool diagram of what's going on with water column transparency that we measure with a Secchi disc, and we have a light meter that we send down the water column. And this is pretty remarkable. We have a huge amount of change here, and the change looks kind of oscillatory. So the depth of the photic zone is varying between 2 and 16 meters over this period of time. So sometimes you can't see down at all. Sometimes you can almost see to the bottom of the lake, crystal clear, down to those beautiful white sands. And we find that transparency, we have watercolor measurements and some VOC measurements, and we find that instead of in most lakes where you see this kind of variation in transparency where it's due to algal blooms going on, this is not the case here. This is an oligotrophic lake, no algal problems. Instead, it's color, it's dissolved organic material that's stained that's causing this change. Well, what is driving that? So, first of all, some of the consequences of that, why that matters so much. This is, again, the same sort of smoothed image of the photic depth, and then behind it is water temperature profiles showing the monomictic nature. So there's the temperature scale. This is a heat isocline thing, and you have every winter the cold water mixing, and then summer stratification setting up, right? And you can see then during the years where the lake is very clear, the water is a lot warmer, and the hypolimnion is really deep and narrow. So yeah, summer hypolimnitic temperatures are warmer during clear years. So this has a huge consequence to the lake. That's the point of that. What is it driven by? Well, what we found is by having a huge long history of hydrologic measurements throughout the watershed, we've been able to relate things like the depth of the photic zone to the amount of water in the watershed at the time, and so we can use the stage of the groundwater, and we see a hugely nice relationship there. And so what's happening is during big stormy times, you get a lot of water moving through the watershed, some of it underground, more of it over land than when the water table is deep, because that water would more naturally percolate if the water table is deep. If it's shallow, it's going to run over land, and it's going to bring color dissolved organic material into the lake from all those wetlands in the watershed. So that's what's going on with this story. And we've been able to relate that very nicely to this major climate driver in Florida, the Atlantic multi-decadal oscillation. We know that matters a lot in the Everglades. It controls about 70% of the water input to Lake Okeechobee. So now all the releases from Lake Okeechobee are regulated according to this cycle. Well, we see the same thing in Lake Annie, where, so this is the record that we know of going back to the 1950s. There's groundwater level superimposed on that, so you can see the tight connection, and there's the photic zone depth, right? Completely driven by that. And one of the cooler things that, well, following on Amanda Quillen, you know, she went out, she was a student in my lab who went out and did a survey of the diatoms of this region and related their abundances and composition to water clarity. I recently took that model and applied it to the sediment core that Bill, you and I took, a freeze core. There's a great story around that I can tell you sometime, but we took this core and I applied her model just recently, and look at that, it's like the coolest thing in the world. It's like going, you can totally reconstruct the AMO, and so the cool thing is we want to go back and reconstruct this for when we, you know, don't have a record of it. So anyway, that's why Lake Annie is cool. We started working in it in trying to figure out what these changes were back in 2005-06, and we decided to have a big workshop of, you know, we invited a bunch of limnologists to try to talk this out, figure out what's going on in it, and at that time, we invited Tim Kratz, who had just been starting up Leon, and he said, hey, this is so cool, because if we put a buoy in this lake, then we'll know that much more about it at a new time scale. So with lots of help from Gleonites, we had a meeting in 2008. We hosted the annual meeting for Gleon, and we invited a whole bunch of people before the meeting to help us do all this. We got the buoy, we got lots of people who knew how to wire all the sensors. Hillary Swain's husband did our mooring system. He's a marine engineer. Putting pressure transducers up, going up the very scary, unless you've had a couple of beers, water tower, and installing the, you know, cell antenna and all that. And then these are all Gleon people who came and did, you know, a whole lot of work, including doing all of our data transmission. You know, they set us up really well with the metadata system, way to transmit data up to Wisconsin. And we learned from that that it takes a village to raise a buoy. We very much need at least a cowboy, a programmer, a director, a limnologist, and an IM specialist over there in the telemetry. And then all of our Gleon friends who helped so much with this. And since then, we've helped a lot of other sites deploy buoys. And what this has done for us is given us a whole new scale to look at, right? Where we can understand now the ways in which short-term variance controls long-term change. So these are the kinds of things that we're investigating in the context of Gleon, now into a little bit of the broader science. The role of episodic events, thresholds, and nonlinear dynamics. You can do that really well if you have high-res data. What's going on with the fate of carbon, coupling physical and biological processes, model development, and site-based things. Those are just some, you know, broader categories of things that we do. We do this in the context of working groups, and so we've been meeting as a larger network all over the world once a year, but then we have these little working groups that break out and we'll go to other places, usually some cabin, and we all just stay together for a week and we write a paper or we do something. And so, you know, face-to-face meetings, we meet a lot over Skype when we're away from each other. We share a lot of data. We've involved all of our students, and a very, very inclusive governance structure, sort of like what we have in FCE, right, where everybody's a part of how the network moves forward. Location of meetings, so I've been to all of them except that one, so it's been a real adventure, and we could do a photo journey at some point, or you can friend me on Facebook, because this is fun stuff that I do on holidays. Okay, these are the different working groups that, or a few of them that currently exist. I've been really active in the climate and lake physics one, even though I'm not somebody who studies physics. The climate story that I told you got me excited about that, and then I've been a part of this theory group. And some of the things that we've done, we've developed tools for making those pretty diagrams of what's going on with thermal dynamics. And that's not just to produce a pretty graphic, but it's to calculate things like the depth of the thermocline and the degree to which wind forcing is changing the depth of the thermocline. Really, really important physical parameters that really have been argued a lot about and how to calculate them. And so this group is full of a lot of physicists who figured this out and found, actually in doing so, a lot of problems with historical ways of calculating very simple things, even like Schmidt stability of the water column. So we now have a program, it's available on the web. I have my undergrads do this in our limnology lab because it's very easy. You just import data sets and you can spit out things like the work of the wind or Schmidt stability or the depth of the thermocline. And pretty graphics to understand change. And that was written up by one of our students, Jordan Reed, who's a lake physicist. And what we've done with Lake Annie is, so here's the long-term record that I already showed you, and here's our high res right now that we put the buoy in 2008, and we can see really cool things like Tropical Storm Faye, not a very big storm, but it came through, rained a lot, and actually sent the mixing depth way down deep, and it continued to be deep the rest of the summer. So one event drove the huge amount of change in the lake. Very cool is in the context of this longer record, our very cold snap in 2010 and then repeated in 2011 caused a cooler hypolimnion. Sorry, that's there. Because it's neat that this hypolimnion stays cold, right? Because it's trapped down there all summer. So one cold week in January can affect the rest of the summer. We would not know that. Had we been sampling monthly, we may have missed that. We've written up the story about Hurricane Faye in the context of a bunch of other lakes that had storms. So as I said, one of the things that we've been caring about in Guyana is the effects of episodic events, because now we can get at that with high-res data. And Eleanor Jennings, she's a professor at Dundalk Institute in Ireland, brought together our data from Annie and a few other lakes to show, and you don't need to get the details of this here, but essentially the arrows are marking events, and we track through the effects of those events through different aspects of, this is like effects of the storm on dissolved oxygen concentration and so forth, and compared that among lakes. So Gleon gives us that nice ability to say, how general are these kinds of responses? Another thing we've done is with Jordan Reed again, the guy who developed Lake Analyzer, we've been able to look across all these lakes of different sizes and shapes at the ways in which wind forcing and convection dynamics influence stability of the water column. This is really core, basic physical limnology that is still open to a lot of questions. And Jordan's work required that we integrate data from over 40 lakes, so it's a lot of processing. We did this in little working groups for, I think it was 3 week-long working groups for 2 years, where we all got together and just took a chunk of these data, and there was a lot of QA/QC, and it was hard to do. But the result was that we could see, here's all our little lakes, and they're not scaled to size, but they're on here according to their little shape. And then this axis is surface area, and that axis is the influence of the wind on mixing. And we see that these little lakes are much less influenced by the wind, and there's like actually a linear relationship here that we didn't know before. This requires lots of data from lots of different lakes to figure out. So again, a student leading a big project and really nailing it. Another student-led project by Chris Solomon resulted in a paper that, if anybody's doing work on ecosystem function and aquatic ecosystems, you probably know this paper that just came out in 2013. It's a really important one. because it builds some new models to understand variance in aquatic metabolism at the daily scale. And we find huge variance in respiration and GPP during the day. We suspected that, but it hadn't been looked at across lots of different lakes. And the cool thing in his work is that he was able to show the degree to which respiration is responding to every magnitude change in GPP. It doesn't have to, but it will in certain settings, especially in oligotrophic lakes and in dystrophic lakes, where that's the main source of carbon that these little guys, algae, are going to use. And so from that paper, we've got figures like this, where here's 40, this is the same 40 lake data set as we used for the other physics paper, and there's Lake Annie up there, and this is essentially just showing, you know, positive values from our dissolved oxygen reading, so that's GPP, and this is R, and you can see in each lake there's a slightly different balance, and this is over like a 2-year period, okay? And from this, he related the degree of respiration, background respiration that's going on, to things like the amount of alloxanous material that's delivered. So now, you know, the more alloxanous material comes in, the more heterotrophic your lake is going to be. So he showed that at the daily scale, and things like, you know, we were able to see that Lake Annie is functioning as a heterotrophic lake, at least in these years, and has a really strong degree of coupling between GPP and R. Not a lot of respiration going on that isn't being driven by the algae in the lake. That's pretty cool, and it's something that would be really, really cool to look at in clear versus stained years in this lake. This is really where I want to go next with this work. And I put, and incidentally, we used, so Greg Tuck, there's Greg, oh, you can't see him, but he's out there with his buoy on one of the ponds down in Taylor Slough. So, you know, he was a student at Dan Childers here. We got him into Gleon, and he started working with Peter Stair from Denmark, who developed some of the initial models for lakes on how to calculate metabolism, whole system metabolism. And Greg applied that same kind of model and was able to show net heterotrophy in these basins down in Taylor Slough. And he makes a strong case in this paper for a great deal of priming going on. So when carbon is really juicy, especially when there's groundwater phosphorus coming in, carbon is really juicy because it's being produced by the algae in the water column, then that primes old organic carbon to be respired. So he'll see in some of these, he has another plot in this paper where he shows where spikes in respiration are following the spikes in GPP and sometimes exceeding them, indicating that this priming is happening. So Greg was really active in Gleon, and I think it made a difference in him being able to take out this paper with a lot of the right people involved to help with the models. So moving on from that, that's some general stuff, Lake Annie, how it fits into Gleon. Where are we going now? A lot of my work, again, has been in the context of this group we call TTG. It's the theory group, but we initially got called that because we were, somehow somebody said we were theoretically terrific. but we don't know if that's true. We haven't published a whole lot out of here yet, but lots of good ideas coming from this group. The main theme of this team is to try to figure out what the controls are on phytoplankton assembly across different lake types to better anticipate when big algal blooms are going to happen, to predict who's going to be in those algal blooms and consequences to diversity. These, again, are really simple, basic limnological questions, but we're learning that what we currently know about these things is very much biased by a historical focus on a certain lake type, deep temperate lakes. And the more we understand about these other lake types around the world, subtropical, tropical, the more we go, oh, wait a minute, you know, some of the things that we thought were true from this sort of narrow focus of our earlier work may need to be reevaluated. So, yeah, some of the questions we're addressing is over what timescales do these things vary? What are the effects of disturbance? Now we can do that with our high-res data, looking at water column disturbance and what happens with the community. And if we have these high-res data, maybe we'll be able to predict change better. There's a lot of talk out here now about leading indicators, right? Where if we see variant structures changing, perhaps that's indicating that a big state change is on its way. Well, you can't do that unless you have some way of sensing that variance at the short time scales. So we're interested in that too. And these are a few members of our team, but you can see there that we're kind of distributed around. And basically the way that Gleon groups are formed is that whoever is interested in that topic goes to the group. And they contribute as much as they feel like contributing. It's really super informal. And in 2010, we kind of established this group and we started meeting annually. And this is our meeting at AVOG. This is the Swiss Federal Institute of Aquatic Science and Technology on Lake Geneva. So that wasn't too shabby to be. But it was in the winter, so it was pretty cool. So again, some reasoning behind what we're doing. And this reasoning is coming from the oceanographic work, and these oceanographers always have like tons more money than limnologists have. And so they've had these sensors out there for longer, and they're able to show from some of these sensor data that things like, here we have mixed layer depth, and you see it changing there in some part of the ocean. And this paper in particular was showing that, anytime you change the depth of the mixed layer, even if it's just momentary, you get a big change in nutrient availability and phytoplankton production. And so, that kind of inspired our work. In fact, this paper ends with a prediction that it's really the 5-day window before an event that enables you to understand that that event's going to happen. So it's whatever's happening, sorry, I'm not explaining this correctly, that if there's a phytoplankton bloom, it's most likely to have been driven by something that's going on immediately prior to it. And if we know what those things are, then maybe we can predict the blooms better. So we started doing some work on Lake Mendota, because there's a really long-term data set available from Lake Mendota up at University of Wisconsin. Emily Cara is a student up there, was a student up there, she just finished up. And again, this was a working group that we just got together up at Trout Lake, spent a week, took all the Mendota data, each of us took a chunk of it, and worked out the QA/QC, got the data streams into a model, and this is essentially just showing that we have the data there and the colored points, and then her model that's projecting the effects of water temperature variability, just one key driver on chlorophyll concentration. And she shows a pretty good fit of a really simple model where phytoplankton abundance is driven primarily by water temperature variance, controlled by the seasonal dynamic in the water column. So from predicting bloom dynamics to predicting changes in assemblage composition, Lizette Senair-Point-Domus is a really big researcher at the Netherlands Institute of Aquatic Ecology in Amsterdam. And she is doing a major revision, or she just published a major revision of the ways in which we think about phytoplankton succession in lakes. And what we thought about it is, as I was saying, like most of our models came from these temperate lakes. She first took those models and revised them. These are just conceptual, literal models of what we should expect should happen in the abundance and seasonal trajectories of phytoplankton and zooplankton in lakes of different trophic status. And then she applied those models to what we know about Arctic lakes and tropical lakes and set up these testable things. So her paper just came out and gave us something to go on to test in these other lake types. So we're excited about that, and she's really stimulating a lot of new ideas and new things that we can test within Glean. And so we're testing those things by taking the high-res data, buoy data with all the sensor information, linking it to medium resolution data, which is the stuff you go out and do monthly, and linking that to even lower resolution data. So the phytoplankton, we got chlorophyll sensors on some lakes. Those are expensive, they're not always accurate, especially in the oligotrophic lakes, not so good. So we're still fairly reliant on count data, microscopic count data, a little bit of a limitation still. We don't quite understand what we're getting from our fluorometers. But here we have the opportunity to link some high-res changes to lower resolution, like classical monthly phytoplankton data. And the steps that we've gone through with this kind of work is to first figure out what's the variance structure in the high resolution data. And this is a typical kind of plot that we kind of look at in order to figure out what are those scales of variance. So it's just a bunch of different lakes and the scales over which key variables are changing. So where it's darker, it's showing a greater degree of effect of that particular time scale. So, we have annual cycles, and for some reason in Lake Annie, a bi-weekly thing going on. And it's sort of interesting to pick these things out and try to figure out what's driving them. And so we're trying to take that variance in the high frequency data and link it to variance in, abundance, for instance, of major algal groups. So this is work that Kaylin Carey is leading up, again on Lake Mendota, where there's a really nice long data set. And this is just showing over 10 years the timescales over which things like diatoms, green algae, and blue-greens are cycling. And it's hard to see the scale, but right around here in each one of these is the annual scale. And the cool thing is over this is a 10-year period. So the neat thing is, why the heck is there not an annual signal? If cyanobacteria, there's one, they're always blooming in the middle of the summer, annual strong signal. But these guys, greens and diatoms, sometimes do, sometimes don't. That's really weird and interesting. It says that no matter what model we come up with, there's probably not a static one to apply to any given lake in any given year. And within these groups, we also know that there's a massive amount of diversity change and assemblage change going on. This is just at the higher levels. So if we understood this within those levels, that would be even more exciting. And that's what we're trying to do with Koji Muraka. Some of you met Koji. He came to visit last spring, and we spent a bunch of time working on this. He's a student at University of Waikato in New Zealand. His dissertation is developing around the classic intermediate disturbance hypothesis, but applying it to high-res disturbances in lakes and to what happens then to phytoplankton communities. And for this project, we're working on 4 lake data sets. That's not very many, but it's the only ones where we could harmonize our, we started out in our Switzerland meeting with like 25 data sets with phytoplankton numbers. And it's so hard to harmonize these data sets, to harmonize the taxonomies. It's really important to do. And to have the same kind of driving variables in all the data sets. So we said, well, let's try it out with these 4 lakes where we know we have stuff in common. And these are some results from Lake Annie where Koji's taken a model. He's kind of a physical knowledgeist too. And he's looking at the accumulation of stratified time. So the more stratified the lake, and the longer it's stratified, the less disturbed you would think it would be. But within any place along that period of stratification, you're going to have variance in stability that's driven by a windy day or a rainy day or an event. And so he said, to what degree can we use, again, just the physical variance and stability to predict species richness based on that unimodal model? So he just took the very simple unimodal model, projected it out for 2 years for each one of these lakes, and the model projections are here in the dark line, and the actual values are in red. This simple, dimple model that is saying that diversity is controlled in a unimodal way by stability. I was blown away by it. I mean, we've always known that water column stability really matters to phytoplankton, but I wouldn't have expected that we would have this great of a correlation with the first try at this. So we're applying this, it's working really well in the other monomictic lake and not as well in the 2 dimictic lakes, and that's what this story is going to be about. We're trying to figure that out. You know, we can have places in there where diversity might be the same, but the assemblage is totally different, and so that's where some of the approaches that I've been using come in, and Coach and I are also working on this. This is just looking at the degree to which similarity in community composition is changing over time. Again, so we're seeing an annual cycle, meaning that communities are more similar if compared to another one exactly a year away from today, right? Makes sense. We can look at that in ordination space and kind of follow the community around, right? And look at the difference between beta diversity, temporal beta diversity, how different the community is now from next time in the winter versus the summer. And this reminds me that this stuff over here is just explaining how this is done. We've been trying to do that in Glen for every project. We do a little PowerPoint describing how each step is done so that somebody else can do it and use it as training. We have little training sessions at the beginning of each meeting where we teach folks how to do it. This is showing the 2 different years that are 2 different years of data are showing different trajectories, but that they come together each winter, which is news for a subtropical lake. And this is pretty cool. This is just showing the effects of water column stability and the metric that Koji calculated, which is the amount of disturbance in the water column 4 days prior to sampling. And these 2 variables, again, among like a whole ton of variables that we entered into this NMDS, these were the only ones that came out significant. And so I built, this is taking paleo models and applying them to the modern setting, but it's something I thought I'd try to see whether or not we could project or predict water column stability and disturbance directly from who's there in the community. And these, again, are really strong models. It's saying, you know, in order to understand phytoplankton dynamics, if we know one thing, the structure of the water column thermally, we can go a long way. So, yeah, again, I've done that on Lake Annie and Lac Faya, and it's less stable for the 2 dimictic lakes, even in the summer. So it's a little weird, and we're trying to figure out why that is. Finally, I want to end with this, that we are getting into some fun work with this. We were frustrated in the analysis that I showed you because I told you we had a bunch of lakes and the data were hard to harmonize, and they were all collected at different intervals and different species, different people counting them, and that's frustrating. So we said over a bunch of Guinness beer at the meeting in Ireland, why don't we all do this together during the spring when the water column is mixing? And we just thought this was the greatest idea. And so we have a bunch of people from lakes all over the world, and there's Emily and Sylvia, and they put a lot of time into this at Lake Annie. And there's a lake in Denmark, and there's Lake Constance or Geneva, and here's Erken in Sweden. And so we all, like, we just committed to it. We're gonna do it. So these are all the people that did it. And we said, let's coordinate this. Try to link water column stability to phytoplankton diversity. Particularly, the idea is that the more structured your water column, the more, this is back to Hutchinson's, you know, paradox of the plankton, the more structure, the more diversity. If we break up that structure, the less diversity. Let's see if that drives succession in the spring to the summer. So these are the lakes that we did this in. And we set up a really common sampling protocol. We all just got on the phone or Skype a whole bunch, set up this sampling protocol. Lake Annie had to go first, so we were the guinea pigs, right? Because our spring happened first last year. And our spring was like the longest spring in the history of record, right, Julia? Julia worked up the thermal data, or the temperature data from here, and it's like, spring just went on and on and on. We said, we'll sample for 8 weeks every 4 days. And poor Sylvia and Emily, and we had a bunch of people helping with this, Anna Sharnickel. We're running up there every 4 days and sampling. And it was really cold for Florida. And anyway, we did all that, and everybody else did too. And New Zealand and Australia, well, they were still going as of our last meeting, you know, because Southern Hemisphere. And then we got a couple more who came on just this spring. And we have this big exchange going on of phytoplankton samples and of microbe samples, and we did zooplankton all the way on up to food web. And Lizette got us a little money to assist with shipping. Otherwise, it's been all volunteer. But it was a lot of fun, and I think we're going to learn a lot out of it. Here's what was going on with the water column of Lake Annie. You know, we kept thinking we were done and started stratifying, and we thought we were done, and oh no, it got cold again. And then we finished when the water column at least had a little bit of a thermal climb. So, yeah, we're trying to get this done. It's hard to do because it's all on volunteer time. So lessons learned, yeah, I know I'm going over it. I'm going to finish this real fast. This is a people network, you know, it's lots of people having fun working together. And what I didn't talk a whole lot about is how much fun we have at these meetings. It's just, we're really close because we get together all the time and we love it and we love working together, and that inspires us to finish up projects and all of that. So, you know, again, it's made up of people who really want to collaborate, so everybody's, you know, usually all on board with how to do stuff fairly and be upfront and open about collaboration and responsibilities and stuff. One of the neater things is that I've shown you a lot of examples of where students have driven projects to fruition. And so we've seen that the students are the main driver of things in Gleon. And so we've tried to build that up as much as possible. And so Kathy Weathers put together a really nice grant, got it funded to support a fellowship program. There's Emily Nodine, she's been a part of that. And the fellowship program is awesome. It's not just teaching people about limnology. It's how to collaborate, how to lead, network science, and all that good stuff that that generation is going to have to be all about. And those kids are from all around the world. And one of our challenges has been how to share data around different places. Like different countries have different, you know, if it's a nationally funded project in China, there's going to be a different, you know, kind of way that might be shared from what we are used to here, where we're mandated to share, you know. So that's been a challenge, and yet, again, the willingness of the collaborators has helped us overcome that, and that's been kind of fun. So, yeah, that's Gleon in a really fast nutshell, and I'd be glad to have any questions, or if people want to get involved, you know, that you can talk to me about it. Thanks.
By Kendra Sherville.
Right.
It really describes what you just exemplified in your collaboration with Leon about building high-performing interdisciplinary research teams and the importance of people networks.
Right.
And
was that the macro systems special issue I just got yesterday? Okay.
Describes what the framework that y'all used.
Yeah, so there's lots of venues for this now, right? There's NEON and there's the macro systems and lots of global ones as well. And so, I think you can find them in your discipline.
But just the sort of non-stratified structure of your team as a promotion of building
that's totally
innovative and inspiring creativity.
I think the fact that our working groups, there is no hierarchy in them, period. Students have just as much, you know, I mean, there's no, and students are leading them very often. And that's so important. And it's cool to watch folks from different cultures sort of either bring new ideas to that or adapt to it. It's very interesting because, you know, I think, you know, I don't want to sound, well, we're getting more used to that and the different collaboratives that I've been involved in the US. And there's other places that are probably doing that even more, but some places that are doing it less. And so it's neat to see, you know, that sort of mind-opening bit, you know, that we all learn from each other's different cultures of how to collaborate. And one of them is just enabling as much energy from students to go from start to finish in their dissertations in that kind of context.
It'll be interesting to follow up on that and watch the relative success, because NEON is the most hierarchical program we have ever put in place. I mean, it has layer upon layer upon layer of hierarchy and stuff. And so it'll be really interesting to see if the results and sort of how the networks change and grow are altered because of that, those different levels of hierarchy.
So this is mostly unfunded, the whole thing is just...
That's the whole, I mean, it is frustrating. There's one RCN grant that came from NSF to get it going in 2006. Research Coordination Network grants are to bring people together to work on a common theme. And that paid for people to go to meetings and to get together and have a little, I mean, we have a program manager type person who has supported a little bit off of that, and that helped. We've had, you know, the fellowship grant, Kathy and Paul Hanson got, and then there's a cyber infrastructure grant that came in to help with some new innovations in transmitting data. And Gordon and Betty Moore Foundation also matched, I believe, the Research Coordination Network grant. That's all within the US. And then in Europe, there's a NetLake program that is a network of European limnologists all around Europe, and all of whom are also in Guyana. And they've brought in a grant that's similar to our RCN grant. And so that's helped quite a bit. So yeah, there's a little money here and there, but it's not like science money. It's to get people together, and then the science is just done, you know, because we like it. Yeah.
And you're like Andy Bowie, on that question, same question, where does the support for that and the telemetry?
It's so sad, I can't take it. Well, so, really, because something awful happened about that last week, but.
Oh, sorry.
No, let's see, you know, Archbold is a private biological station, and so they, Hillary has cared a lot about the lake, Hillary Swain, the director, and so she pretty much threw the money into the buoy system that we were told was gonna cost $10,000 and ended up costing $35,000. That's just for installation and getting it going, and we've had to replace sensors, a lot of them every year. And we just go up, we do it on like Saturdays, you know? I mean, the labor is just, it's all volunteer. Yeah, so I'm trying to get money for this unsuccessfully. So. Yeah.
I'm glad
Hey, Evelyn?
Oh, sorry.
Yeah, oh, sorry,
Hello?
we always forget about you guys up there. Can I take that one?
Of course.
Yeah. Yeah.
Hey,
Rudolph.
this is Rudolph. I'm kind of embarrassed to even ask you because I started working with you on this years ago and really never finished what we started. But I would like to get back to this, and I'll come and talk to you sometime. But can you tell us a little bit about the variations in DOC concentrations throughout the year, and then how they vary with depth on a seasonal basis, or do they vary with depth on a seasonal basis?
I do not know the answer to part B because we have not sampled in a depth specific way.
Okay.
We're just taking surface samples, and the variance across the year depends on the seasonality of our rainfall. So typically concentrations go up to maybe 5, 6, 7 parts per million in like August, September. In a
Okay.
really, really rainy year, they might go over 10. And then they drop back down after winter mixing. So there's some indication that, when the lake mixes in the winter, that there might be some buildup down there, and that it comes back, that the refractory stuff comes back up into the water column. And then
Okay.
the lake
Yeah,
clears
so-
back out in the spring and summer.
Okay, because I was wondering if it's mostly groundwater fed, if you accumulate CDOM at the bottom, then when it mixes, it will actually be photoreactive.
Oh, cool.
And with some of the more juicy, I mean, there could be some priming happening during that time of the year, so.
I like that thought. We should look at it. I know that we did do the comparison between groundwater DOC and the lake water DOC, and it was absolutely the same. Not quality, but quantity. But it would be interesting to look at the quality. The sand
Yeah.
just is so transmissive. It's like the water outside the sand is the same inside the
Okay,
sand.
and I'm buying a probe so that can be lowered at different levels, so for CDOM, so we could quickly check the depth profiles for the CDOM.
Cool.
So,
Wow.
I'll talk to you about it.
Definitely. That's awesome. It would go a long way to that hypothesis about priming.
All right, I'll talk to you about it, thanks.
Thank you.
One more question?
Is there time?
One question?
Okay, well, I'm really glad you did the lessons learned, and on the data side, I was just wondering, I mean, you talked about like the having to harmonize and the issues of data sharing, but as far as analyses, have you found that, especially with the high res, is it requiring kind of new types of analyses? Is it more filling in the gaps? What
Yeah.
lessons have you?
Right. So there's some tools that Glenn has developed for just taking data streams and cleaning them up that have been really, really helpful, because we were doing that fairly manually for a while, and it was crazy. So these people in New Zealand built a really nice tool for that. And then the programs that we're using for the wavelet analysis and for your stuff is, that's out there, but it's nice to use that technology for this application. I think we're learning more about how to look for correlated timescales of variance. And so I didn't have any clue about any of that mathematics and approach to understanding biology before I worked with Leon. So I'm learning a lot about that. It may not be new though to anybody else, but I'll definitely making some advancements in the application of those kinds of time series analysis.